if we can just go back to the title slide, because I changed the title on the talk, and I did that because I decided to make the title the question at hand. And that is, how does HIV persist in infected individuals despite antiretroviral therapy, which is called ART, and I'm sure you all know that, but I'll just call it ART probably throughout the talk. And we want to know the answer to this question, of course, because we need to understand how it is that the virus is persisting in patients if we want to cure it. We have to know what it is we're going after, basically. What's the mechanism that HIV is using to persist so that we can attack that mechanism, sort of target that mechanism to eradicate it. So we can go on to the next slide now. So as you all, back one to slide 2. So as you all know, when antiretroviral therapy became available in the mid-90s, the number of deaths from AIDS declined, and so did the number of diagnosed cases. At that time, it was predicted by some that ART would eradicate HIV infection, that it would be the cure to HIV infection. And they thought that because what ART does is it blocks the replication of the virus, which means no new cells can be infected. So if ART is successful, then all of those cells that were already infected before starting treatment should eventually die off. So when you start ART, it should prevent any new cells from getting infected, and then over time, some cells that were already infected would die within days, some would die within months, some might take, a few years, but if you stayed on ART long enough, eventually all those cells should die off and it should be the cure for HIV infection. And as you all know, that's not what happened. There are patients on ART for 20 years or more now, and they're still HIV infected. And so we want to understand how it is possible that these cells are persisting all these years, or is it that ART is not really doing its job fully? In other words, maybe there's still low-level ongoing cycles of replication occurring during treatment. So you sort of have this, constant reinfection of new cells during treatment that's preventing ART from being a cure to infection. And so that's the question that we're trying to address, is what's the mechanism that HIV is using to persist during treatment. So we can go on to the next slide now, slide 3. So as you all know, when infected individuals initiate ART, their level of viremia in the blood, so the number of virus particles in the blood, declines about 4 logs. And it gets down to very, very low levels. On average, just about 1 virus particle per mil of blood plasma. And that's shown here in red on this graph. These are sort of the low-level virus or viremia that persists in patients on treatment. So you can measure very, very low levels of virus in the blood on treatment. But the number of infected cells, and that's the blue line at the top there, the number of infected cells actually only declines about 1 log. So despite the fact that the amount of virus in the blood goes down to extremely low levels, the number of infected cells persists in patients on average about 200 or so infected cells per million peripheral blood mononuclear cell, or PBMC. So that's a really high number of infected cells, probably too high for these infected cells to be persisting through ongoing viral replication. That would be an awful lot of newly infected cells every day. And so we suspect that these cells are persisting through a different mechanism, and that's part of what our studies have investigated. Okay, so we can go on to the next slide, slide 4. Okay, and as you all know, so that red text that popped up at the bottom of slide 3, as you all know, if you stop treatment, the level of viremia rebounds right back up to where it was prior to starting treatment. And that can happen either from the development of resistance or from stopping therapy. So we know that these infected cells that are persisting on ART are capable of reseeding the infection if you stop treatment. Okay, so slide 4. So how does HIV persist during ART? And there are 2 schools of thought. One is that it persists because ART is not fully effective. So you still have ongoing cycles of viral replication despite treatment. That's one school of thought. And so that would mean that if that's the case, that HIV is persisting because ART is not fully potent, then that means that if we develop more potent ART, so that would be ART that better is able to penetrate the tissues, get into every single possible infected cell, and block the infection then of any new cells. If we could do that, then we could cure HIV infection. And that's what some people say, is that HIV persists through ongoing cycles of replication. And the other school of thought is that ART is fully effective. So it is blocking the infection of new cells, at least to the level that it would be the mechanism that's supporting all of these infected cells that are persisting during treatment. And in this school of thought, it's the cells that were infected before starting treatment that are persisting throughout treatment, or at least many of them are persisting. And we believe that they're persisting, and there's lots of data to show this, that they're persisting through clonal proliferation. So it may not be that that cell that was infected before starting treatment lives for 20 years, but rather that it lives long enough, maybe a few months or so, to be able to undergo cellular division. And when it undergoes cellular division, it makes a copy of that, of the integrated provirus, or the virus, the HIV genome with it. So even when that cell dies, it's already been replaced by a clone of itself with that provirus. And so that's the second school of thought. So we set out to determine which of these is likely the case. And so on slide 5, okay. So in slide 5, I'm just going to go ahead and jump right to what the answer is with this summary slide for the community that was sent to me by Linda. So the question is, does HIV continue to replicate during antiretroviral therapy? So in other words, is that the mechanism that HIV is persisting despite treatment? Is it persisting through ongoing cycles of replication? And what we found is no evidence for HIV replication, either in the blood or in the lymph nodes during ART. So this suggests that ongoing cycles of viral replication is not the mechanism that HIV is persisting during treatment. This means then that developing more potent ART will not cure HIV infection, because more potent ART would just further block the infection of new cells. And we didn't find evidence for infection of new cells, at least not at a level that would be needed to maintain the HIV reservoir or allow HIV to persist during treatment. So therefore, we must develop treatments to eliminate long-lived cells and proliferating cells that are infected with HIV if we're to cure HIV infection. And the good news is,
can I interrupt? There is somebody either eating, or can you, can everybody just mute their speakers or their phones? There's somebody like chewing and eating in the background. I'm so sorry. Thank you.
Okay. Okay, so the next bullet point then is that, so like I said, we have to develop treatment to eliminate these long-lived cells or these proliferating. And the good news is that there are new approaches already underway to try to target and kill HIV-infected cells. So what ART does is it blocks the infection of new cells, but new therapies are attempting to target the infected cell itself and kill the infected cell itself. And so possibly combining these approaches, one targeting and killing HIV-infected cells, and 2, preventing the virus from going on to infect any more cells. Maybe combining those would be the key to curing HIV infection. So, okay, so just a little background here and a model for viral replication on ART. So first, let me just tell you, without ART, you have virus particles that infect CD4 positive T cells. The viral RNA is then copied into proviral DNA. That DNA is then integrated into the host genome, and it now becomes a part of the genome of that cell. So that cell can now generate new virus particles, which can go on to infect new cells. When this happens, the virus makes errors, or the enzyme, one of the enzymes for the virus makes errors in copying the genetic information. So in every cycle of replication, when the new provirus is integrated into the newly infected cell, it contains genetic information that is slightly different from the round of replication before it. So after many years of this, with cycles of newly infected cells, the virus accumulates a very high genetic diversity. This is called HIV evolution. So we can detect viral replication because we can see the accumulation of these genetic mutations over time. We can see the virus becoming divergent over time from the virus in previous months or years. So we can use this then as a tool to determine if viral replication is occurring. So meanwhile—
Can I ask a question?
Yes, sure.
So it's not clonal expansion, right?
So that is what I just described is viral replication.
No, but I'm saying you said that you eliminated one of the potential 2 reasons for ongoing virus in your study, and you found it was not clonal expansion. Is that right?
No, actually, so what we found is we didn't find any evidence for viral replication during ART. We did find evidence for cellular clonal expansion.
Okay, gotcha.
Yeah.
Thank you.
And so, so what I'm showing here is actually both viral replication, and that's what I just described, where you get this accumulation of genetic mutations in the virus over time, leading to this very high genetic diversity of the viral population. But what I hadn't quite gotten to yet, but what you just mentioned, is that while this is ongoing without treatment, we also have infected cells that are proliferating or dividing. And those cells can sometimes also make virus particles. So without treatment, there are 2 sources of viremia. One is ongoing cycles of viral replication infecting new cells every day. The other is cells that were infected in previous days or months or even years that have undergone cellular proliferation, and some of those can also make virus particles.
So
now,
treatment,
sorry?
Mary, is that before the cell proliferation? Is that so? These cells, did they develop before treatment, or?
Yes,
Okay.
yes. And what we found is, so you have cells that are infected that are undergoing clonal expansion, both before treatment and during treatment. And that's what we have found. So because all ART can do is block the infection of new cells. It can't do anything to cellular division. So if a cell is already infected, that cell can divide, making many, many copies of the provirus that's integrated into that host genome, and ART can't do anything to block that. So it happens both before treatment and it continues to happen during treatment, is what we found. And so, so if we want to investigate this question of, do we have ongoing cycles of viral replication even during ART? If we want to investigate this question, we can do an experiment. And that is to start therapy very early after the transmission of the virus in individuals. And when we do that, if we start treatment, if those individuals are sustained on treatment for 5 years, 10 years or more, then we can sequence the virus and we can see, does it match the virus that was there before they started treatment? Or did the virus accumulate all of that diversity that we would expect to see the virus accumulate if there's ongoing replication, just like in patients who are not treated? And so we did that experiment. So in a cohort of children who were infected at birth, treatment was initiated shortly after birth, when that virus was still very, very homogeneous. So it had virtually no diversity. So they started treatment then. We looked then when the children were 10 years old, and we sequenced the virus when the children were 10 years old. And if there was ongoing cycles of viral replication, what we would expect to find is a phylogenetic tree that's shown on this slide that's up right now, slide 6. So the pre-therapy virus, so each of the symbols on this phylogenetic tree are representing a single virus particle. So as you can see from pre-ART, before starting treatment, all of these symbols are stacked right on top of each other. That means those are identical sequences. So in this case, the virus had no genetic diversity at all. All the sequences are identical, which is what you expect in a very early infection. So now if we were to sample 10 years later, if there's ongoing virus replication, what we would expect to see is for the tree to look like these red symbols here. And that horizontal distance that you're seeing between those symbols, that's the genetic distance between each of those virus particles. So what this tree is telling us is that before treatment, all of the sequences are identical from the virus, and then 10 years later, now they're all very diverse. They're all different from each other. This is what the tree would look like if there was ongoing replication during ART. So now we can go on to slide number 7, and I'm just going to show you what the tree would look like if there was no viral replication. So the first tree is showing pre-therapy, now in red. Sorry, I reversed the colors. That was a mistake. So the first, so in pre-therapy, the sequences are all identical. If there's no viral replication, and now we sample 10 years later on ART, we still expect those sequences to be identical. We don't expect to see the accumulation of any mutations. This is different now from the tree on the right, which is showing what we would see if there was viral replication. So that's what we're looking for. So we did this experiment in children, and we looked to see, if we sample before treatment and then we sample 10 years later, do we get a tree like the one on the left, which shows no viral replication, or do we get a tree like the one on the right that's showing that there is viral replication during that 10 years of treatment? So now on the next slide, I'll give you an example of what we found. But first, I'm going to show you what we found in a child who was not fully suppressed on ART. So this is a positive control for this experiment. I just wanted to make sure that if a patient is not fully suppressed on ART, are we actually able to see the virus diverging over time? And that's what this is showing. So this is a child who, for the first 1.5 years on therapy, was not suppressed, had detectable viremia for that first 1.5 years on therapy. Then the child was able to be suppressed and was suppressed for about 6 years. So in red are the sequences from pre-ART, and as you can see, there's a lot of identical sequences there. There are some that have a few nucleotide changes. So this child had already started to accumulate a little bit of diversity, even shortly after birth. And then when we looked at the virus when the child was 9 years old, keeping in mind that this child had not been suppressed for the first 1.5 years, we can see that the virus evolved over that time. So this is evidence of viral replication in a child where we would expect that there was viral replication. So this is just our control, showing that we can use this method to detect viral replication if it's there. So the next.
And with clonal expansion, how come there's no diversity mutations?
So the clonal expansion, when a cell undergoes clonal expansion, it's copied by the host enzyme, which really doesn't make errors, or very, very rarely makes errors. So when you have clonal expansion, you don't see this divergence of the virus over time, because it's making a exact clone of the cell that was infected before treatment. So I haven't shown any of that here, but I will show some of that in later slides of the evidence of clonal expansion. Okay, so
The point of this is, Mary, that this child that wasn't fully suppressed has all of this different divergence because the virus was not fully suppressed all that time.
exactly, right. And so I just did that because lots of times people will say, well, how do you know your test is sensitive enough? Maybe you're just not seeing it. And so, you know, we had the opportunity to get samples from children who were not fully suppressed, just to show that when we do this analysis, we can detect the viral replication if it's there. And so that's the purpose of this child. And then I'll show you an example of some children who were fully suppressed for the duration, and you'll see the difference in the trees. So if we can move on. Okay, so what just popped up on this slide is just some statistical tests to see if the sequences on ART did diverge from the pre-therapy. And we do that because we don't want to say, oh, we can look at this tree and we can see the difference. We want to actually apply some, you know, rigorous statistics to it and ask, is it really different? And what these data are showing you is that they are statistically significantly different. So the virus is, we can detect replication of the virus when people are not fully suppressed on ART. So let's go on to the next slide. Okay, so now this is a child who was fully suppressed for the duration.
Wow.
Did we jump ahead a slide or no? Okay. So this child initiated ART at 1.8 months old, and we obtained a sample from just before starting therapy, and then a sample from 8 years on ART. And when you look at this tree, you see something very different than the previous tree. I think we actually skipped a slide. Did we skip one? Yeah. Oh, no, we didn't. I'm sorry, we didn't. Okay. Okay. So in this child, you can see that the sequences from 8 years old, they're shown in black, in the black triangles, are not on longer branches. The population has not diverged from what was there in pre-therapy. So in pre-therapy, almost all of the sequences are identical. We find a few that have a few nucleotide changes. That's diversity that occurred in the virus before starting ART. When we look now 8 years later, we find that the sequences, most of the sequences are identical, and there are a few that are a little bit genetically diverse. That diversity is not statistically significantly different than the diversity that was already there before starting treatment. And so what this tells us is that for 8 years, there was no evidence for virus replication in this child. And the next slide just shows an example of another child. We did this in a total of 10 children, and the result was the same. I'm just going to show you these two. This paper is published if you're interested in seeing the other 8 of them. It was published in JCI, Journal of Clinical Investigations, last year. So this child, we collected a sample shortly after starting ART, 9 months on ART, and a sample 8 years later. And we asked, did the virus diverge over that 8 years? And what we found is that the virus did not diverge. So the sequences in pre-therapy, most of them are identical. There are some that had accumulated a little bit of genetic diversity. And then when we look after 8 years, The virus population, again, still most of the sequences are identical, with a few that have accumulated, that before therapy accumulated some diversity that we already detected in the pre-therapy sample. So what this tells us is that using these methods where we can detect viral replication when we know it's occurring, using these methods, we were not able to detect any virus replication in patients who are fully suppressed on ART. And so this suggested to us that HIV is using a different mechanism to persist in patients on treatment. So without treatment, the virus is infecting new cells every single day. It appears that ART prevents that from happening, but HIV is using something different then to persist in patients. And that different thing, we believe, is clonal expansion, and there's a lot of data to support that. And so if we move on to the next slide.
Also a huge argument for early treatment.
Yes, it is. Yes. Before the virus has accumulated so much diversity, yeah, it may be easier to target infected cells where the virus is genetically, has very low diversity, than to target infected cells with viruses that have very, very high diversity. So after then addressing that first question and deciding that viral replication is unlikely to be the source, to be the mechanism that HIV is using to persist during treatment, we did another experiment. And this experiment was addressed at this question of clonal expansion. So if HIV isn't persisting through viral replication, is it persisting through clonal expansion of cells that were infected before treatment? So to do this experiment, to investigate this question, one of the things we did was to initiate treatment in individuals not in early infection when the diversity is very low, but instead to look at samples from patients who initiated treatment in chronic infection when their viral diversity was already very, very high. And if we do this and collect a sample right before starting treatment, we can sequence the virus particles, and they're from pre-therapy, they're shown on black in this tree, and we find that the virus population is highly diverse. That's, of course, because the virus has been replicating for many years before starting treatment. But if the virus is persisting through clonal expansion, and we now sequence the virus in the blood after years of ART in these individuals, we might expect that the population becomes homogeneous, because those cells that are persisting during ART are undergoing clonal expansion, making many, many copies, identical copies of the same provirus. So if some of these clones, cellular clones, are able to generate virus particles, all those virus particles would be identical because they're coming from a population of cells that have identical proviruses. So we did this experiment, and I think on the next slide, I'll show you some results. So first, I'm just going to show you the blood. So if we can just back up for a minute on these slides and just look in the blood. So, so we first just looked in the blood. So we sequenced the virus from a patient who initiated treatment in chronic infection, and we obtained a sample. These are samples from the DARE Collaboratory. They were shared with us by Steve Deeks. So this is a patient who initiated treatment in chronic infection, and we were able to get a sample from prior to initiating treatment. And the viral diversity in this individual is shown in the open triangles on this tree. So as you can see, most of these these proviruses in these cells in this case, are genetically distinct. There are a few that are identical. Right at the top of the tree, you see two that are identical there. So then we collected a sample, or Steve, Rebecca Hope collected a sample for us 5 years later. And we looked again in the blood cells. And as you can see, the population went from this very diverse population to a lot of identical sequences. Those identical sequences, we believed, were from a cell that was infected with that provirus before treatment, and that cell had divided and divided and divided, making many, many copies of that provirus. And so this tree looks exactly like what we would expect to see if HIV is persisting through clonal expansion. So after this, what we did is we went back to this patient sample, and we actually sequenced the site within the host genome that the HIV integrated into. Because if indeed these proviral sequences are from a clonal population of cells, then they should all have the identical site of integration of that provirus. And that's what we found.
Wow.
So these are from all of those identical sequences in the closed black triangles are the result of clonal expansion. And so we believe this is evidence that HIV is persisting during ART through clonal expansion and not through viral replication. So, so then we decided to look in the lymph node, because as you all probably know, in the lymph node is where people say, well, maybe there's no ongoing viral replication in the blood, but in these tissue sanctuaries where maybe ART isn't able to penetrate well, maybe there's still ongoing cycles of viral replication there in the lymph node. And so if that were the case, then what we would expect is that even though in the blood, the virus population did not become divergent, in fact, it is persisting through clonal expansion, what we would expect to see in the lymph node, if it was a different mechanism that HIV was persisting, and if that mechanism was viral replication, then what we would expect to see in the lymph node is that the viral population would have become very diverse, even more diverse from pre-therapy in the lymph node. And it would be very different. Sorry.
But you do see some diversity in the lymph node there, right?
So you don't see any more diversity than what was already there before treatment. So in the open triangles are the pre-therapy, and as you can see, there's a lot of diversity there in pre-therapy. And then when we look on therapy, and this is 5 years on therapy, you see a lot of these identical sequences. These are from an expanded clone. And yes, you see some sequences in the lymph node that are diverse. But as you can see, those sequences are not on longer branches on the tree than those that were already there in pre-therapy. And so what that tells us is that you have a lot of cells that were infected before treatment that are persisting and undergoing clonal expansion. Because if after 5 years, if the mechanism was ongoing viral replication, then after 5 years, there would be a lot more diversity than what was there in pre-therapy. So the tree, in other words, would look like we saw in that baby or that child that was not suppressed for the full time on ART. And remember how we saw the sequences get on very long branches? That's what you would see. And that's not what we see here. So the next question we asked is, so we see this large expanded clone in the blood, and we see this large expanded clone in the lymph node. So then we asked, are these the same clone? And if they are the same clone, then that would tell us that the cells are trafficking freely between the blood and the lymph node. And so that's shown on the next slide. So this is now taking the sequences on therapy in the blood and the sequences on therapy in the lymph node and putting them on the same tree. And what you see is that that large clone that was in the blood matches that large clone that was in the lymph node. So this is a single cell that was infected before treatment. And that cell has undergone massive cellular division. And Now there are many, many cells that have identical proviruses through this cellular division, and those cells are freely trafficking between the blood and the lymph node. And as you can see at the top of the tree, you see these sequences that are more diverse, and that diversity is not different in the blood and in the lymph node. They have the same diversity. It was also not different from the pre-therapy diversity, if you remember from the previous slide. And again, some of the sequences in the lymph node and the blood match, indicating that there are other smaller clones in this individual. So we have this one very large expanded clone at this point in time, and we have another population of a smaller clone. And so what Bob Silicano and some others have shown is that these expanded clones kind of, they expand and then they contract, and then maybe they'll expand again and then they'll contract again. And so you might see different sized clones at different time points. So this tells us that the virus is persisting through cellular proliferation rather than viral replication.
Do we know why that is, Mary, that there's different at some points in time than others?
No, we don't know that, but it's likely because your T cells of your immune system are meant to respond to an antigen and then to proliferate massively in response to that antigen in order to attack it and get rid of it. And so we suspect that these large clones probably, maybe someone got infected with a cold virus or the flu, or who knows what. And so it just happens to be a T cell that's responding to that, and that T cell happens to be HIV infected. And so the clone might get very large while it's sort of attacking this, adenovirus or rhinovirus or whatever the person happens to be infected with. Those are just cold viruses, and then it would contract again once that virus has been eliminated. So it may be just responding to something. And there may be other reasons as well, but that's just one possibility.
So Mary, you're saying that the goal then of cure or eradication should be to target the clones?
Yes. Yes.
And how do we do that?
Well, cellular division, it sounds a lot like cancer therapy to me. I don't know much about that, but if we want to target proliferating cells, there certainly are therapies to do that. But I think this is much more difficult than cancer. The difference with cancer is that cancer cells are different from your normal healthy cells. And so it's easier to target them because there's something different about them from normal cells to target. Whereas these cells are probably, other than being HIV infected, are the same as normal cells. So the difficulty would be targeting these and killing them without killing all of your normal healthy cells. That's the challenge.
Well, and also cancer.
Okay, so Nelson.
I was just going to say.
No way.
Good.
Go ahead.
No, go ahead.
So
Is there no way to target the cells based on their integrated proviruses?
Well, that would be the hope, and that is exactly what a lot of people are doing. So what they're doing is trying to activate these proviruses to get them to make a lot of virus, and that would create then the signal, from normal cells. And not only might the virus that it's producing kill the cells, but the immune system might actually kill those cells if we can sort of awaken those proviruses and get them to make a lot of virus. And that's what a lot of people are doing, and that's the kick and kill strategy. So target the cell after activating the provirus so that there is something very different about them from normal cells. And that would be the hope, is that can be done.
So far, kick and kill isn't working.
No, it's not working so far. but it's possible that combining different latency reversing agents, or of course, we hope for better and better latency reversing agents over time. It's possible that it can get better.
So question here. So we, in 30 some years, we have never found any phenotypic differences between an infected cell and a non-infected cell, like on the surface or receptor wise. So there's no way to identify these cells, like using any methodology besides just trying to activate them to reproduce and produce more provirus.
No, there have been a lot of claims, but like the what was it, the CD32A, or the there have been a lot of claims that there are some things that are different about infected cells that from uninfected cells, but I don't know that any of those have really panned out to be something that could be exploited towards a cure.
Thanks.
So there are still people searching for markers like that. And, there are papers coming out, every few months with another possible marker for HIV-infected cells. And so possibly one of those markers will turn out to be something that can be exploited and used to target infected cells.
You seem to raise some doubts about cancer drugs because cancer cells are different. And also, what about the safety issue of cancer drugs?
Well, I don't know enough about cancer to have an informed opinion on it, but what I do know is that there have been an awful lot of HIV-infected people with cancer who have undergone chemotherapy, and it doesn't appear to have made much of a difference in their in the size of the reservoir, their HIV reservoir. Although I don't know how sort of thorough those studies have been, and I haven't read them really carefully. But to my understanding, current chemotherapy hasn't seemed to have an effect on the HIV reservoir.
No, I mean toxicities.
Yeah, well, that's the problem, Jules. It's not efficacious, and it's too toxic. So it's like a double whammy. I mean, we've yet to find something that is effective and is also not toxic. So it's not that the strategy stinks, it's that the drugs we have so far haven't been effective. So
yeah,
right.
Yeah, but there are newer cancer treatments all the time, immunotherapy and CAR T cells and all sorts of things that may be useful in designing strategies towards curing HIV, and of course people are attempting those approaches as well. So I do think it's possible that we could make a dent in the reservoir by combining some type of cancer therapy with ART, and maybe even with latency reversing agents. So the next slide, 14. Okay, so now to sort of prove that these identical sequences that we see are clones, that we see the same clones in the blood as we do in the lymph node, which suggests that HIV is persisting in the blood and in the lymph node with this using the same mechanism. And that mechanism is clonal expansion of infected cells and not viral replication. So just a little bit of background here, and I think I already said all this, but just to say it again. So HIV can integrate into many sites throughout the human genome. So if an infected cell clonally expands, all the viral DNA in that clone will have the exact same integration site. So we can identify these clones by sequencing the integration site of the HIV. And if we find the same integration site over and over and over again, we know that's an expanded clone, because the human genome is 3 billion base pairs. So the chance that you're going to see the exact same location of a provirus being integrated over and over again, by chance is highly, highly unlikely. So it has to be that that was a single integration event and that that cell underwent clonal or cellular division. So we can use this integration site data then to show that HIV-infected cells clonally expand. So we did that in these patients in the lymph node and in the blood, and that's on the next slide. So what we did is we sequenced the sites of integration in the lymph node and in the PBMCs, that's the blood cells. So we obtained 580 integration sites from this one particular patient from the lymph node. We obtained 247 integration sites from blood cells. We found a total of 40 expanded clones, that is 40 populations of cells with sets of identical integration sites. 16 of those clones were found both in the lymph node and in the PBMC. So we know that these cells are trafficking freely then between the lymph node and the PBMC. 17 of the clones were found only in the lymph node and 7 were found only in the PBMC. So we applied some statistics to this. So if you just hit the forward, yeah, we applied some statistics to this. So we said, are these populations of clones different in the lymph node and in the blood? Because we sampled twice as many integration sites in the lymph node, we have to account for that. We have more chances to see clones in the lymph node because we look twice as hard there. So if you normalize for the number of integration sites that we obtained in the lymph node and in the blood, it turns out that the number of clones and the types of clones in the lymph node and in the blood are the same. So they were not statistically significantly different. So it just appears that HIV is persisting both in the blood and in the lymph node through clonal expansion of cells that were infected before treatment and not through viral replication, the infection of new cells.
So Mary, was that just one lymph node you found 580 integration sites, or was it more than one?
That was one lymph node.
Wow.
Yeah. Yep.
Okay.
Yes, there are a lot of infected cells persisting in patients who are on ART and have been suppressed on ART for many, many years, which is our first indication that HIV is not persisting through viral replication. Because so if you have, let's see, so you have about 1 billion new HIV-infected cells a day without ART. That only declines after many, many years on ART. That only declines to about 100 million. So the amount of, so you persist at about 100 million infected cells on ART. So for the mechanism to be ongoing viral replication, to sustain that, the amount of evolution that you would see after many years would be tremendous. But yet what we see is that the virus population has not diverged from pre-therapy at all. And so it suggests that that sort of 100 million cells throughout the body that are infected during ART are persisting through cellular division. So it's just making copies and copies and copies of the same proviruses that were there before starting treatment. Yeah. And these expanded clones can contain replication-competent proviruses. So you all may know the story from Ya-Chi Ho and Katie Bruner, where they showed that the majority of HIV proviruses are defective. They can't even make virus. We don't really care about those. It's only 1 or 2% of the infected cells that are persisting during treatment that can even make intact virus particles that are able to go on to infect new cells. Those are the ones we care about. And unfortunately, these expanded clones can contain replication-competent proviruses, and that was published a couple of years ago. And there have been some papers since then that have confirmed that result in other patients as well. So the next slide. Okay, so then we ask the question, I'm getting down to the end here now. So then we ask the question, how can these clones survive when they're infected? When the cell is infected, it's supposed to be recognized by the immune system and killed by the immune system. So how are the cells able to survive despite infection, and not only survive, but divide and divide and divide and make many, many copies of themselves? So we suspected that that was because the majority of the cells within these expanded clones are latently infected. That means the provirus is integrated, but it's sitting there completely dormant. It's not making any RNA, it's not making any virus particles. And so the immune system doesn't know they're infected. That's what we hypothesized. So this slide is showing these little evil guys. Those are infected cells. And so if some of them are making RNA, we suspect that those are recognized by the immune system and killed. But it doesn't matter, because those that are latently infected just keep on dividing and dividing. And so you sort of always have this source of virus, but you have the majority of the cells within these expanded clones that are completely quiescent, with completely quiescent proviruses that are hiding from the immune system. And so we suspected that that was what was happening. So we did another experiment, and that's on the next slide, to see if this is the case. And we published this method that we developed last year. So what we did is we were able to obtain single cells from within these expanded clones. They all have identical proviruses. That's shown here by the black proviruses, all being the same color. These are identical proviruses because these are all members of the same clone. So we developed an assay where we can see which ones are latently infected and which ones are actively transcribing and making virus particles. So if you can just click forward. In this example, it's showing 2 of the cells within this expanded clone that are making virus particles, while the other 6 are completely, the proviruses are completely dormant, latent, not doing a thing. And that's how these cells are able to survive. This is what we hypothesized, and this is what we developed an assay to test for. So in this example, if you click forward, we would report that 25% of the cells within this clone are actively expressing HIV RNA and making virus particles, while 75% of the cells within this clone are latently infected. And that, therefore, they can just hide from the immune system, and they can continue to divide and continue to divide. So you always have this small fraction of them making virus, so you always have this low-level viremia on art. But the majority of them are just completely silent. And so that majority then can go on to divide and divide and divide, because they're not going to be seen by the immune system. They're not going to be killed. And so we did an experiment using samples from an individual, as on the next slide, where we looked at 34 different expanded clones. So on the x-axis, you have 34 clones. 3 of them are in red. That's the first 3. We gave those names because those we know have the ability to make replication competent viruses. These are proviruses that are intact. These are that 1 or 2% that Bob Silicano, Yachi Ho, and Katie Bruner have talked about. These are the ones that really, really matter. The other ones are either known to be defective or are probably defective, but we just didn't identify what the specific defects were. But it's these 3 that are the most sort of important, these 3 clones. So we used that assay that I told you about, and we looked at cells from within these clones and said, what % are making virus? And that's what's shown in these bars. So for this first clone, that's the AMB1 clone, we call it, it was only 2%. So of this clone, and by the way, we looked at 1,200 cells with this identical provirus from this clone, 2% of them were generating virus particles, and the other 98% were latently infected, not making any virus at all. The provirus is completely dormant, and they're hiding from the immune system. So that means 98% of them can just go on dividing, and the immune system will never know they're there. Only 2% are actually making virus. And so if you stop treatment, it's that 2% that results in that virus coming right back. And so for the other clones that we looked at, the fractions were very, very similar, very small fraction that are, generating virus particles or proteins. There were a few clones where the fraction was higher, but for the most part, on average, it's only about 4% of the proviruses that are actively making virus particles, while 95% are dormant, and that is how these cells are able to survive despite treatment, despite your immune systems. And this is the mechanism then that HIV is using to persist. So basically, it takes cells get infected, the virus, the provirus remains dormant, and the cells just divide, and that appears to be the mechanism that HIV is using to persist during treatment rather than ongoing viral replication. So
let me ask you a question. You say that it hides, the latent virus hides from the immune system. So with things like zoster and CMV and other things that are dormant in many systems, isn't it the immune system that keeps them dormant in those cases, or is it the same sort of hiding process?
So I don't know enough about the herpes viruses to know the answer to that, but I do think you're right. So, but with HIV, what I do know about is that it appears that the viruses, so cells get infected, and it appears, although nobody really knows the answer to this question, to be honest, but it appears that by chance, when that provirus integrates, some of them just become dormant. And those are actually selected for, because the ones where the provirus integrates and makes lots of virus particles, of course, those get killed off by the immune system. So it sort of selects for this population of proviruses that are not making virus particles. So in that way, yes, the immune system is responsible for it, but only because it's killing those that are making virus particles, leaving those behind that are latently infected because it doesn't see them.
It just seems like a different process than the other viruses.
I don't know the answer to that because I just don't know enough about the other viruses, to be honest. And I don't think we know enough about HIV latency to be able to say that.
Okay.
But I will say that I did talk to someone who studies, I think it was herpes simplex, and she said that the fractions are about the same. So most cells are latently infected, and you have a very small fraction of cells that are in that lytic cycle. And that's always the case. So when there's a herpes outbreak, it's not that those fractions change, it's that the amount of cells in those that have this lytic infection, the amount of virus that's produced from those cells that are in that lytic cycle just is increased.
Uh-huh. So it makes the exact thing then.
Yeah.
Yeah, okay, thanks.
It might be similar. I don't know. Okay, so I think the next slide summarizes everything. Yeah. So just to summarize, we found no evidence for viral replication in the blood or the lymph nodes during HIV treatment. Now, that doesn't mean that there aren't absolutely any new infected cells during ART. What it does mean is that it's probably very rare, and it's highly unlikely to be the mechanism that HIV is able to persist despite treatment. The reservoir includes populations of infected cells that persist and proliferate despite infection and despite treatment, because most of these proviruses are latent. Curing HIV infection, therefore, must include targeting and eradicating populations of proliferating infected cell clones, much like targeting cancer cells. And so the next slide, I think, is my acknowledgements. Yes, and there are many, many people on here because these studies are always a huge collaborative effort. UCSF, Steve Deeks, Rebecca Ho, Jeff Milush, and others have been so wonderful with the DARE Collaboratory sharing samples for us to do these studies, and they're wonderful, amazing people to work with. So I really want to thank them. The baby studies that I showed you were in collaboration with folks at Stellenbosch University in South Africa, who are also an amazing group of researchers. I have a lot of help and consulting with John Mellors, the University of Pittsburgh, John Coffin from Tufts University, and of course the folks in my lab in the TRU, who did all of this work, and other folks in our program, the HIV-DRP, Dynamics Replication Program, who have given me much advice on this. And Leidos are the folks that did the integration studies with us. And of course, the patients, we always have to thank the patients. It's amazing the generosity of them to share the samples with us for us to be able to address these questions. And so lastly, this slide I put in because I think it was Linda who asked if I would put it in, because I put this slide up when I presented some of this data. I think it was at the Persistence meeting. It was in Miami because it's Christmas time. And so I had my Christmas tree, which I dedicated to all of the women in 2017 and 18 who spoke up about sexual harassment and gave everybody a voice and to recognize them and the Me Too movement.
Go, Lady Mary. That's what I said. Wonderful. Wow. I'll tell you now, I remember why I wanted you to do this. What an amazing set of studies. I mean,
Yeah.
so clear and comprehensive.
Thank you.
Wow. Do we have any last questions for Mary? I'm hesitant to make her stay any longer because it's after 7 and she has no air conditioning where she is. So
it's fine. It's fine. I would have done this from home where I have air conditioning, except for my dogs are too badly behaved, and I knew they would just bark in the background the whole time. So it's my fault. I'm here doing it. I don't train my dogs properly.
Yeah, I have a quick question. I don't know if Michael just posted a study that they're doing in Seattle with mycophenolate. Are you involved, or do you know a little bit on the, that's an organ rejection drug, right?
Oh yeah, no, I don't. I'm not involved, and I don't know about that study actually.
Okay.
Okay. Well, why don't we let Mary go? Well, I think we asked you all of our questions while you were presenting this, and thank you too for the painstaking way that you explained all this really complicated stuff. I mean, it's really great.
Excellent.
Oh, thank you. Thank you.
Lady, that Christmas tree, it's my favorite.
Well, I'll put it up again this Christmas, so. Yes, it's my favorite Christmas tree ever.
All right, thanks everybody too for participating.
Thanks a lot.
Thank you.
Bye-bye.
Thanks.
Thanks.
Bye.
Bye.
Linda, if you'd stay on for a moment.